Hi, my name is Christian Lee. I'm a graduate student at the University of Toronto in the Department of Medical Biophysics. I'm a member of the Ryman Lab working at the Ontario Institute for Cancer Research. And the title of my talk is Investigating Regional Somatic Mutation Rate Variation Across Functional Elements in Whole Cancer Genomes. So tumors evolve through the accumulation of somatic mutations. This is nicely visualized by this familiar diagram shown here. So initially, we have a normal cell that over time acquires driver mutations and eventually develops hallmark properties of cancer, such as sustained proliferation, avoiding cell death, and so on. These now tumor cells can give rise to various subclones and even distant metastases. But the bulk of mutations are actually passengers, those that don't confer cell select growth advantages and play a more passive role in oncogenesis. However, studying passenger mutations can still be very informative of mutational processes, genomic instability, genome evolution, and even inform driver discovery and more. So it is known that somatic mutation rates are highly variable across genomic scales. For example, at the domain or megabase scale, mutation rates correlate with properties like replication timing and chromatin accessibility. So taking a look at this diagram, on the y-axis we have the relative mutation rate, and then various genomic features throughout. So this portion here, underlined in gray, corresponds to late replicating and dense chromatin. As you can see, there's a relatively higher mutation rate in this region compared to these other regions at this large megabase scale. At the gene level, mutation rates correlate with properties like transcription level and various epigenetic modifications. So shown in green is a specific histone methylation mark associated with an increase in transcription but a decrease in relative mutation rates. And so when looking at the subgene level, mutation rates correlate with specific transcription factor binding. Famously, CTCF and cohesin binding sites show elevated somatic mutation rates. And so this is visualized by these very sharp and narrow red peaks throughout the diagram. At the finest scale, it has been shown that various mutational processes sleep behind specific signatures characterized by various proportions of single base substitutions in their surrounding nucleotide contexts. For example, SBS3 is indicative of defective homologous DNA damage repair. And so for this talk, we are interested in the subgene level, looking at mutation rates affecting functional elements in the range of tens to thousands of base pairs. And so this brings me to the overall hypothesis that regional mutational processes act on functional non-coding elements in cancer genomes. Most mutations are likely passengers, while a subset may affect gene regulatory networks, chromatin architecture, and drive cancer. And so the first aim is concerned with overall mutation patterns and processes across entire classes of sites. So first, we wanted to create a pan-cancer map of regional mutation variations. And to do so, we first developed a tool in R package, and then set out to validate and refine CTCF mutation enrichment with that tool. Then, we wanted to extend this to other regulatory elements and regions, including transcription start sites and open chromatin. Additionally, we'd like to focus on the transcriptional and pathway context of some of the findings from above. So in order to carry out these aims, we used the following data and workflow. We started with 120,000 CTCF binding sites from ENCODE, composed of 70 different cell lines. Later, we looked at 38,000 transcription start sites from Ensemble, and over 560,000 attack-seq peaks derived from 410 TCGA primary tumors. Our mutation dataset includes somatic SNVs and indels from the pan-cancer analysis of whole genomes. This includes over 2,500 tumors from 25 different cancer cohorts, each of which contain 25 samples or more. So then we then feed a single class of these sites, as well as these somatic mutations, into our newly developed tool, Regression Models for Regional Mutations, or RM2 for short. And so RM2 works by stacking sites of the same class and calculating the number of trinucleotide context mutations in sites compared to their flanking regions. So this is shown here, where the dark gray regions are the sites of interest, all of the same class, and the light gray regions are the immediately flanking regions of the same length. The different colored X's represent different mutations in their trinucleotide context. And so then these mutation counts are then used to construct a new data frame like the one shown here, where we have a column for the mutation context, its position, its count, and an indicator of whether these mutations occur within the sites or the flanking regions. Additionally, RM2 incorporates a mega-based mutation rate estimate based on an adjustable number of bins. By default, and shown here, this was set to 10. And so in these cases, the 8,500 mutation falls into bin 3, whereas the 12,700 mutation falls into bin 7. And so the rationale is that together, by including mutation context as well as mega-based mutation rate bins, this allows us to account for base pair and mega-based scale variation while isolating these site-level effects that we're interested in. And so more specifically, RM2 uses negative binomial regression to evaluate whether we can explain significantly more the variation in the mutation counts when including site membership as a covariate. As a readout, RM2 indicates whether there's a pattern of mutation enrichment, depletion, or no difference across our aggregated sites of interest compared to their flanking regions. And so this is an overview of the RM2 results applied to the CTCF binding sites. The different columns show different cancer types, with pan-cancer shown on the far left. The y-axis is split into 4 different panels by mutational properties. The circles correspond to the FDR-corrected p-values, whereas the colors correspond to the fold change. And so directing your attention to the very top, the first row confirms the CTCF binding site mutation enrichment that we discussed in the introduction. And so this is very strong in pan-cancer, as well as esophageal adenocarcinoma, liver hepatocellular carcinoma, as well as stomach adenocarcinoma. And so the second panel subdivides mutations into select mutation signatures. I would like to highlight SBS17A and SBS17B, as these show very pronounced signal in pan-cancer, esophageal, and stomach adenocarcinoma. And so the figure on the left shows a distribution of SBS17B mutations within CTCF binding sites in esophageal adenocarcinoma. Here, the x-axis is the relative position to the midpoint of CTCF binding sites, and the y-axis is the mutation count. The red bars fall within positions within the sites, whereas the gray bars fall within the flanking regions. And so this clearly visualizes the enrichment of SBS17B mutations within the sites compared to the neighboring regions. And so 17A and 17B are of largely unknown etiology. However, some recent reports have connected them to oxidative damage. 17A is driven by T to C single base substitutions, whereas 17B is driven by T to G single base substitutions, both of which were captured here in that third panel. Finally, at the bottom, we consider the strand, either Watson or Crick, as well as the transcription direction, either forward, reverse, bidirectional, or none. And so an interesting result is that the intergenic mutations, so on both the Watson and the Crick, so both the sense and the antisense strand in these non-transcribed regions, show an enrichment of mutations, which is particularly strong in pan-cancer, again, esophageal, liver, and stomach adenocarcinoma. And so this is just an overview and confirmation of CTCF binding site mutation patterns and enrichments. We showcase how RM2 can refine previously documented patterns by analyzing these additional subclasses of mutations. And so in the previous slide, we looked at how various types of mutations accumulated within all CTCF binding sites. But we can also easily look at how mutation patterns accumulate across subdivisions of sites. For example, we were interested in the relation between mutation rates and CTCF binding site conservation. On the left, I've plotted the distribution of site counts on the y-axis against the number of cell lines on the x-axis. We group CTCF binding sites into 5 equally sized bins based on how many cell lines a given site was derived from, where 1 is the minimum and 70 is the maximum. The 5 different groups are shown by these 5 different colors, where bin 5 corresponds to these highly shared sites across cell lines. And so the idea is that this is somewhat a proxy of CTCF binding site conservation and activation. So then we separately fed these 5 groups of CTCF binding sites into RM2 and produced the results shown here on the right. And so here we see 11 different plots for different cancer types. Along the top, we see pan-cancer, esophageal, liver, and stomach. But at the bottom, we capture additional cancer types that weren't shown in the previous slide. And so again, in all these plots, the x-axis is the relative position, whereas the y-axis is the number of mutations per megabase per patient. The different colors correspond to the different bins, matching the diagram on the left. And so again, bin 5, shown in blue, are these highly shared sites across cell lines. Additionally, the solid lines correspond to those with an FDR less than 0.05. In addition, there's also a red line in all these panels, which corresponds to all CTCF sites examined together. And so this allows us to easily compare these different bin level effects to all sites combined. And then the last point here is that these different numbers along the bottom correspond to the FDR corrected p-value taken from the most significant bin, which in all cases was from bin 5. And so the prevailing pattern across all these different cancer types is that the blue lines, so bin 5, shows a very sharp enrichment of mutations within the sites compared to the flanking regions. The other bins, bins 1 to 4, are relatively flat for all these different cancer types. And for example, if we focus on esophagus adenocarcinoma, what we see here is that bins 1 to 4 are relatively flat, whereas the red line corresponding to all CTCF sites shows a modest enrichment. But when you only look at bin 5, these highly shared sites, you see a very dramatic enrichment within the CTCF binding sites and a depletion in the flanking regions. And so this suggests that the CTCF binding sites highly shared across cell lines are actually driving this mutation enrichment. And so what is particularly interesting is that research has shown that the more conserved CTCF sites are more functionally important, meaning disruption of these highly conserved sites can have more severe consequences, which are often related to changes in chromatin architecture. And so with the remaining time, I'd like to switch gears and discuss the application of RM2 towards other functional elements, namely transcription start sites and attack-seq, which is a measure of open chromatin. Additionally, we've incorporated some matched mRNA data as a proxy of transcriptional activity because transcription itself is a source of genomic instability. Then we also like to examine the functional context of transcription start site mutations, enrichment of transcription start site mutations using pathway analysis. So these are results from applying RM2 to both transcription start sites as well as open chromatin or attack-seq sites. The x-axis is broken into 6 different columns based on mRNA abundance, where the far left corresponds to genes that are silent, and the far right bins are those highly transcribed genes. And so across several different cancer types, the trend appears that transcription start sites of more highly transcribed genes show an enrichment of mutations. This is particularly strong in melanoma as well as pan-cancer. However, when looking at attack-seq sites, the pattern is more scattered, where we see some weaker and modest mutation enrichments in the open chromatin regions of more highly transcribed genes. We also see some depletions when looking at the open chromatin regions of silent genes, or those lowly expressed genes, as seen here in melanoma. And so this suggests that perhaps this mutational enrichment in highly expressed genes is promoter-specific and less active in other functional sites that just fall within these general regions of open chromatin. However, because there are about 14 times more attack-seq sites than transcription start sites, there's a huge difference in power, and so perhaps these differences are better visualized by these plots shown on the right. And so here we can nicely see the different mutation rates across bins. We have 5 different columns here corresponding to different cancer types. The top row shows the results for transcription start sites, the middle for attack-seq, and the bottom shows the mRNA abundance in these different bins, which show nice separation. The different color lines correspond to the different bins, where green corresponds to silent genes. And so in all cases, for both transcription start sites and attack-seq, these dark green lines are at the very top, and they are relatively flat. And so this provides support to the idea that non-transcribed regions of the genome accumulate more mutations than regions that are actively transcribed. So as we move towards the bins containing highly transcribed genes, so those shown in orange and red, some patterns begin to emerge, and we see some strong patterns of mutation enrichment within the sites compared to the flanking regions. This is more pronounced in the transcription start sites than in the attack-seq sites. For example, this is particularly strong in skin melanoma, which has been characterized by recurrent promoter mutations. So as we can see, the highly transcribed bins, the orange and the red, show very sharp peaks, whereas the other bins, shown in green and in lime green, show no pattern. And so when comparing this to the attack-seek sites, all of the sites here are flat. And so overall, these results indicate that transcription start sites or promoter regions show an enrichment of mutations in a select group of cancers, particularly at those sites attached to highly transcribed genes. Furthermore, the signal is much weaker when we broadly consider all associated regions of open chromatin. And then finally, to examine the functional context of these patterns of transcription start site mutation rates, we performed an RM2 pathway-based analysis where we considered transcription start sites connected to genes of established pathways. So this allowed us to identify processes that contain strong transcription start site mutation enrichments and ask if they are biologically relevant. And so ultimately, we were able to isolate 372 different pathways, which are shown here. The different colors correspond to different cancer types, most of which are melanoma. And so we were able to isolate several housekeeping pathways like RNA processing, ribosome biogenesis, but we were also able to identify some cancer-related pathways like apoptosis and DNA repair. And so this is interesting because we isolated these pathways when considering enrichment of non-coding mutations at transcription start sites, most of which are passengers. And so a conservative interpretation would be that these transcription start site specific mutation enrichments in genes of cancer-related pathways are simply footprints of genomic instability, while the other extreme could suggest a more active role for these non-coding passenger mutations that instead contribute to driver mechanisms and tumor heterogeneity by affecting gene regulation. And so just to summarize, we constructed RM2, a statistical model to identify regional mutational processes. With this tool, we confirmed CTCF binding site mutation enrichment and further refined it both in a mutation and site context. For example, we looked at the enrichment of SBS 17A and 17B in specific cancer types, as well as we showed that shared CTCF binding sites across cell lines were really driving this enrichment signal. Switching gears, we showed that transcription start site specific mutation enrichments correlate with mRNA abundance of target genes. However, the signal is much weaker or even lost in widespread regions of open chromatin. And then finally, a pathway-based analysis of transcription start site specific enrichments isolated some cancer-related pathways. And again, this was captured largely with passenger mutations enriched at transcription start sites, suggesting perhaps these non-coding mutations contribute to or even harbor some driver mechanisms of cancer. And so with that, I'd like to thank my lab with special thanks to Dr. Ryman, Dr. Abrabo, as well as Oliver. And then these are just a couple of links to the tools that I talked about today in the presentation. And thank you for listening.